ACLF, by definition, is acute-on-chronic liver failure and is the development of liver failure in an acute phase on a patient that already has a chronic liver disease, which is cirrhosis. So acute and chronic liver failure is different than acute liver failure because acute liver failure, for example, from poisoning or drug hepatotoxicity, it develops in a healthy liver. In this case, in ACLF, you have already a sick liver. And ACLF has an extra hepatic component, which means that not only the liver fails, but also you have renal, circulatory, lung, brain failure, and also you can include in that failures muscle failure because they have sarcopenia, they have intestine failure because they have gut translocation, and also they can have an important immune dysfunction. Systemic inflammation is the major driver of ACLF, of the development of especially extrahepatic organ failures in these patients. So systemic inflammation is very intense in these patients. It's at the same level to the level of patients with sepsis. We performed a study 5 years ago in which demonstrated that the levels of cytokines, which are markers of inflammation, were even higher in patients with ACLF than in patients with sepsis. The reason why these patients have this so intense systemic inflammation is not properly known at present, but we think it is because the presence of PAMs and DAMs in the circulation of these patients. The abundance of PAMs in the circulation of these patients is secondary to the impaired permeability in the intestine. That's why it's so important the gut failure in these patients, because it allows the translocation of these pathogen-derived PAMs that stimulate the immune cells. On the other hand, we have an increased production of DAMs. DAMs are damaging, molecules that are released during the damaging of the cells. And in the liver, you have cell death, and this release a lot of DAMs, such for example mitochondrial DNA or ATP. Our group investigates why these patients have systemic inflammation. What are the mechanisms? First, we focus our interest on the release of PAMs and DAMs and how these molecules bind through the toll-like receptors to the immune cells, activating these cells. Then we also investigate, once these cells are activated, how they manage to have what is called immunometabolic status, in which they use a lot of energy because systemic inflammation is highly cost. It has a high cost for the patient because you have to maintain these cells and to generate a lot of anabolic reactions and also to produce proteins, cytokines, and other chemotactic factors. So inflammation is a very energy costly process. There is this concept of immunopathology, which means that cytokines are not just inflammatory markers, but they also trigger tissue damage. So cytokines have the ability to induce necrotic and apoptotic effects in cells and of the tissues, and therefore they impair the viability of these cells, leading to tissue dysfunction or organ failure. In addition, inflammation also affects the mitochondria, which are the powerful energy producers of the cells, and therefore mitochondria is also contributing to the cell damage and tissue damage in these patients. Inflammation and immunosuppression are two sides of the same coin. So you have a hyperinflammatory condition that actually takes all the energy for these cells to carry out the defensive properties against pathogens. So on one side you have an excessive activity of these immune cells, but the host defense properties of these cells are impaired. For example, they do not degranulate well, they do not phagocytose well, they do not produce radical oxygen species, enough radical oxygen species, and all these properties are key for defending yourself against the pathogens, meaning bacteria or fungi. Our group basically studies lipids, and in particular lipid mediators. So lipid mediators are small lipids that are bioactive, and they are generated from polyunsaturated fatty acid, for example, arachidonic acid. And these lipid mediators are potent pro-inflammatory factors. For example, when you take an aspirin, you are inhibiting these lipid mediators. So our major interest is on the role of these lipid mediators in systemic inflammation in patients with ACLF. The research is oriented towards the role of mitochondria. So mitochondria are the main producers of energy in the cell, but if damaged, they release, for example, cytochrome C, which induces the activation of caspases, and then caspases induce programmed cell death. And then this cell death contributes to the release of the so-called DAMs, these molecules that activate the immune cell. So together we have a triad of inflammation, cell death, and mitochondrial damage that are closely connected. The most important contribution is at the clinical level. Actually, was the characterization of ACLF as a new syndrome 10 years ago. But on the translational side, we have contributed first describing that systemic inflammation is the major driver of ACLF development in patients with decompensated cirrhosis. Second, as I said, mitochondria play a major role, and this was actually the result of a translational project initiated in the laboratory 3 years ago. And third, we are now starting to understand how mitochondrial damage induces cell death in, for example, in liver cells. And this is a cycle, a vicious cycle of cell damage, cell death, then release of mitochondria-derived DAMs, and also then the activation of the immune system in response to these DAMs. One of the major limitations at this moment is the availability of a proper experimental model of ACLF. We have some models, but we need to refine them. These models, for example, are good models of liver failure, but not reproduce all the liver failures present in the patients. Some of these models only have liver and renal failure, but they, for example, they do not develop circulatory or lung failure. So this is the major challenge at this moment, to translate the basic discoveries into therapeutic targets. Of course, Having a good experimental model is key, but at the more mechanistic level, we need to understand how the immune system works in these patients. So the immune system is the most important factor involved in the development of organ failures, especially those that are extrahepatic. At this moment, we don't know why these patients, they have neutrophilia, which is increased number of neutrophils in the circulation, in the context of lymphopenia, which means there's a low number of lymphocytes in the circulation of these patients. So we don't know how we can correct this. We need to find a way to get a balance between the immune system, especially a balance between the innate immune system and the adaptive immune system. So we need to not only fight the inflammation, but also immunosuppression. For example, this low count of lymphocytes. So we have performed some studies with B lymphocytes, and at this moment we have some data indicating that normalizing the number of B cells can protect these patients from infection.